In general, please, you can interrupt me at any time. I'm really happy if this is going to be more of an interactive session. Personally, I always hate it when people hold hour-long monologues in these meetings. So if there's any question at all or any comment from your side, yeah, feel free to just interrupt me and ask. Right, so my idea for this talk was to talk a little bit about data science and medicine in general, and kind of what I think are good principles to follow, like what I learned across my projects, how to successfully make a data science project in a medical topic work. And obviously I'm going to use my main one of my main projects as the example. In that project, I'm trying to identify drivers of a certain autoimmune disease. Right. I think we all know by now probably that data science is becoming more and more important in medicine, because there's just more and more data being produced. And of course, that means there's a huge potential for gaining additional knowledge, developing new therapies, and so on by using this data. But it also means that biologists really need to learn new techniques and new ways of working and thinking to deal with this. Yeah, and I find it a super interesting intersection of fields. Right. So what is this project that I'm going to use as my example? So I'm part of a consortium, and the goal of this consortium is to fight an autoimmune disease called vasculitis. And those of you who've had Latin in school will have already translated this as inflammation of vessels. So it's an autoimmune disease where the immune system starts to inflame blood vessels, and that can have very severe and very different consequences depending on which vessel is targeted. And in the subtype of disease that I'm focusing on, the immune system mainly targets large blood vessels, and very, very typical is that it targets this temporal artery that you have on the side of the face. And that means that the symptoms that people have got is usually they have very strong headaches, and they turn blind because the blood supply to the eye is not sufficient anymore quite quickly. So that's why you really quickly want to try to treat them. So you want to be able to diagnose them as quickly as possible and as accurately as possible. And that is often very, very difficult at the moment. And also you would really like to offer them a more specific treatment, but at the moment, everything that is possible is you can give them steroids to really bring down the whole immune system in a way to stop this reaction from happening. But since the mechanisms behind this disease are still not very well known, it's very hard to develop more specific therapies and also more specific diagnoses. Right. So the way that people do get diagnosed usually is that they take a biopsy of the inflamed vessel, and then you can see one histological image here. And even as someone who's not familiar with histological images, you see if this is supposed to be a blood vessel and it has this like tiny little hole in the middle, it doesn't look quite right. So this is really, really swollen. You find all kinds of unusual cell types in this like swollen blood vessel wall. And one cell type that you find there are T cells, which you wouldn't usually expect in this tissue, definitely not in high concentrations. So that makes them suspicious. Why are they there? What do these immune cells do there? And are there maybe drivers of this disease? Do they have anything to do with the mechanism, how this disease works? And that's kind of my research question. That's how we started. And the theory would be that maybe there is what's called a self-antigen. So some component of the blood vessel wall that serves as an antigen that the T cells react to, that they bind to with their receptor, and that triggers this immune response. That's just a theory at this moment. whether there is such an self-antigen, but that would be kind of the ultimate goal to find out if that is, and kind of what component of this blood vessel wall is it that triggers this immune response. The data that I'm working with is T-cell receptor repertoire sequencing. So we have these T cells, and each of them has a T-cell receptor on its surface. And you see a T-cell receptor here in blue on the side. And these T-cell receptors very selectively bind to epitopes. And you see an epitope here, just like a small molecular structure in the middle. That's just a short peptide. Usually that would be some peptide from a virus or a bacterium, but in case of an autoimmune disease, it's going to be from some protein, a part of a protein that's in this blood vessel wall, for example. And there's this very, very specific interaction between the T-cell receptor and the epitope. And my goal would be to find the T cells, the T-cell receptors that are typical for this disease, for vasculitis. And then we can find out maybe, okay, what are they binding to? What is this little component that triggers them? Just to give you all the kind of vocabulary that we're going to need, if we look at all the different T-cell receptors that we find in a patient, and there's going to be millions of different ones in each of us, that is called the repertoire. So if I'm talking about repertoires, I mean thousands and millions of T-cell receptors that we find within one patient, and then each T-cell or T-cell clone has one T-cell receptor. And just to like literally show you what the data looks like, I just took a little screenshot of one of the data sets. So I'm going to have clone count. So how often did they find this specific receptor in the sample? What fraction of the total receptors is this? And then some amino acid sequence. I'm going to talk about these like gene families here in a little bit later, because for the first things that we're going to do, you don't need to worry about them yet. Right, so that's kind of the biological background you need to understand my project. So let's start with the data science now. So first of all, of course, you always need your patient samples, but you also always want to compare them to something, right? So you also need controls. Don't worry about the abbreviations. GCA is just the abbreviation for the subtype of vasculitis that we're looking at. So just replace GCA with vasculitis, this autoimmune disease in your head. We've got data from 25 subjects, and we've two different data types. We've got blood samples, and then we got these tissue samples from the biopsies that they took for diagnosis. And that's quite interesting, because then we can kind of compare what receptors are found just circulating and which receptors are found really localized at the site of the inflammation. And then we got some controls. And what I find very interesting is we have healthy controls, but we also have controls of patients with a different autoimmune disease. It's called PNH. We're not going to talk about it, but it's also an autoimmune disease. So it's kind of, these people aren't healthy, but they're a different kind of sick. So that's also interesting to kind of maybe filter out the patterns that maybe aren't so specific to this autoimmune disease. To gain an additional like larger data set as controls, I also downloaded this publicly available data set of more than 600 healthy controls. That's good because it's big, but of course the in-house controls are still very, very nice, because they were really done with the same methods in the same lab with the exact same experimental setup. Right. So one of the first things that I think we should always do if we start a data science project is to find some way to check the quality of our data. And this will be vastly different depending on what kind of data you're working with. But what I did for my project is doing rarefaction curves. Rarefaction curves try to answer the question, did we, during the sequencing process, during the experiment, did we even catch all the clones that are there? Right? So there's going to be, I don't know, 1 million clones in your sample. How many of them did we even find? And what you're plotting is how many sequences do you find versus how many unique sequences do you have? And basically, if the line is flat, that means if you add 1,000 sequences, you basically haven't found any new ones. So you've seen them all before, so you very likely have found all the sequences already. And if the line's still going up, that means you were still in the process of finding new sequences when you stopped. So there's likely to be sequences missing. And you can see that most of the samples that we have are quite good, but also not perfect, I would say, especially here on the right. These are the blood samples. You can see that there's still, like, these lines are still growing. So that means that we have to expect that we're still missing some of the clones that would be there, which is good to know.
Can I ask something shortly?
Yeah, absolutely.
What happened to the lines which stopped? I mean, shouldn't that theoretically just flatten out as the others?
I mean, ideally they would, but it's just that the sequencing process isn't always perfect. That's exactly what I'm checking here, right? You would hope that you would always get to the point where you've sequenced everything that's there, but you're never going to get there, right? And then in some samples, it worked well, and it's very flattened out, like let's say this green one, and sometimes it didn't work very well, like this blue one, and there were probably far more sequences there, but experimentally you just didn't manage to catch them all for some reason. Biology is messy, experiments are messy. It's not always going to be working out perfectly. But basically, if you do such an analysis and you see that none of your curves have really leveled out, that should worry you. Then maybe you can ask some collaborators whether they could maybe repeat the experiment or repeat the sequencing, or yeah, that will just tell you that the quality isn't very high, and then yeah, that it will just make the analysis that you do a little bit less reliable, right? Because then basically, if I'm saying like, I'm not finding this clone in this person, I can't reliably say, because it's not there, that it could very well be, yeah, because we haven't sequenced properly.
So you see on the x-axis, you see the number of new sequences you find, but the line ends when the number of reads stops.
Exactly, exactly. Okay. You also see that we don't have the same number of reads for every sample, right? Like there can be huge differences there as well. Yeah. Exactly. Right. Good. Thank you for the question. And then the next thing we want to do with our new wonderful data set is we want to try to get an overview. In this case, I also really wanted to check is the distribution of like very simple, potentially confounding variables like age and gender similar in my control samples compared to the patients that I'm interested in. And one thing that I immediately found was not a surprise is that my GCA patients are just significantly older than the control group, because GCA, this type of vasculitis, very typically occurs in people over 60. Yeah, so we see immediately that the age distribution really isn't the same. And we're going to plot whether that has an influence on some of the factors in a moment, but that it's just something that it's important to know about this and to keep in mind that this can confound our analysis. And also for the males to female ratio, GCA is a disease that occurs more frequently in females than in males. That means that the distribution isn't 50-50 in our data either. And actually, for the healthy controls, it also was disbalanced for just luck reasons, I guess. But in the PNH control sample, for example, and also in this additional publicly available data that I downloaded, it's rather 50-50. So that also means that there could be some gender-related effects that that can confound our analysis if we're looking at whole repertoires. And now I want to show you something where there's probably already an age-related influence that we're seeing. So one very simple analysis that what you can check is how many different clones do we find in each of these samples. And how many different clones, that's also a measure of the diversity of your immune system, right? So if you have many different T cell clones, many different T cell receptors, that means that basically your immune system can respond to a large number of different threats. And it's already known that this decreases with age. So with age, there's going to be less flexibility, less diversity in your immune system, less different clones, which we're actually really seeing here in the GCA patients. And so this is an effect that we're observing that I think is very likely to be traced to age. However, what we also see is that in the biopsy, the number of clones is even more significantly lower, like a lot lower compared to blood. All the other three are blood samples, right? So this, I think, is not age-related anymore. This is probably an effect of that in this tissue, in this vascular blood vessel wall tissue, usually you wouldn't really find any T cells at all, or very, very few. And so it's not a big wonder that we're not finding huge numbers of clones invading. And just to check kind of my theory that what we're seeing, this effect of like lower diversity, lower number of clones in GCA, that it's an age effect. I just calculated the correlation coefficient between age and two different diversity measures. So Cow2 and Shannon diversity, just two ways of measuring diversity of repertoires. And we see that they're clearly anti-correlated to age. So basically, the higher the age of the subject, the lower the diversity. And that's what you would expect from literature. But that also means that diversity measures aren't really going to be useful in our analysis, because they are very much influenced by age, and we have this age imbalance between the GCA patients and the healthy controls. So all the effects that we might be finding there, we can never really trace them to the disease. They're all like, there's always this high likelihood that it's just related to age. That's just, just an example of like where you can do some analysis, but not always, the effects that you're observing may not always be traced back to the disease as you would like it, but can sometimes be the result of some confounding variables. Right, so let's disregard diversity and look at other things instead. So one thing that was really interesting to see was that you have far fewer clones and far more expanded clones in the GCA biopsy. So an expanded clone is one that makes up more than 2.5% of all the sequences that we find. And that means that when we look at the GCA biopsy samples, we really sometimes find very, very few different clones. And that means we can plot them as these kind of cake diagrams. And you see that there's samples where really 3/4 of the sequence that we find, it's just this one clone. Or where maybe we just find 4 different sequences and then some that occur very, very infrequently. That means that I think all the T cell receptor sequences that we find in these biopsy samples, all the receptors, all the T cells that are really at the site of the inflammation, they're quite suspicious, right? They might be involved, and there aren't really that many of them. So that's interesting. That makes them far easier to look at and to analyze. Right. So next thing I did was I wanted to find out whether there are patterns that we find in these T cell receptor repertoires. So in the whole of the T cell receptors that we find in these patients. Basically, I was wondering, could a very, very simple model distinguish the patients from the controls? This is another recommendation that I have, is always start with the simplest model you can think of, because if that's already working well, you don't need to think about more complex models. And also that already gives you an idea of how clear of a signal are you expecting. So this was the simplest model that I could come up with. So I'm just showing the model. I'm giving it a subset of disease samples and a number of control samples. And then I'm just asking it to look at the overlap of the test samples, so test repertoire, with the disease and with the control samples. And if it finds more T cell receptors that the test sample has in common with disease than the control, then it's supposed to predict that this is also a diseased patient. And if the patient has more sequences in common with the controls than the disease, then it's supposed to predict that it's also a control sample. So very, very, very simple, just looking at identical sequences in the repertoires. And I got a perfect result. So it seems that it's really, really simple to classify the patients based on overlapping sequences. So that already gave me the first hint that, okay, apparently there's a quite clear signal in these T cell receptor repertoires that this very simple model is picking up on, which was really good. I was really happy at the time to see this, because when we started the project, we weren't really sure whether the T cell receptors and the T cells are involved in the disease, or maybe they're just a byproduct. They don't really have anything to do with the disease itself. And in that case, they probably wouldn't have been specific. There would just be some random sequences there, right? But what we're finding, this is for the biopsy samples, is that really, there seems to be a large overlap between the sequences that we find in these patient biopsies compared to all the controls. Right, so now we know there's a signal there. A simple model can pick up on it. Now we want to find the signal. We want to identify the T cell receptors that are the signal, that are critical, that are suspicious to be involved in this disease. So I defined some criteria. The first is the T cell receptor is supposed to be expanded in the biopsy. So I really want to look at those receptors that we find in the biopsy samples at the site of the inflammation, because I think that makes it more likely that the receptor is really involved in the disease. Then, of course, I want it to be shared by patients. If just a single patient exhibits this receptor, then I don't think it's a major factor in the disease. I want to look at those that the people have in common. And then thirdly, I think the suspicious receptors are those that are disease-specific, so that we don't find in patients without the disease. And with all these, applying all these three criteria, I identified 49 unique T cell receptors that fulfill all three criteria out of millions that I have in a sample in total, right? So it's a really small number. Yes? Oh, was there a question? Or just some background noise? Okay, background noise, I think, right. Okay, right. And then when I looked at how these sequences are distributed, I wasn't surprised anymore that the model was picking up on this very easily, because I found that every patient has at least 5 of these suspicious clones in the biopsy, and some of them had like 17 or 18 of them. So out of just a bit less than 50, there were patients who had almost half of these suspicious sequences in their biopsy. So that means, yeah, it was quite likely to that the model could find these in the overlap, right? And also, if you check these sequences with models that calculate the generation probability, so how likely is a receptor to even be produced in the first place? How likely is it to be there by random chance? And then we also find that the suspicious receptors are less likely to be there just by random chance, just because they have a high generation probability compared to the unsuspicious ones. So that means they are rarer than average in general, but they occur with very high frequency in our patients. So that's quite a clear signal. No wonder that the model picked up on that. Right. So the next part, we're going to look more closely at these receptors, at the sequences that we're finding. And so we need to do a little bit more biology at this stage to be able to understand the following analysis. Right. So these T cell receptors they are produced in a very ingenious way, because if your body is supposed to produce millions of different receptors that are all similar but have different specificities, that's quite a difficult task. And if all of these receptors were just hard-coded in the DNA, that would take a lot of space, and it would also mean that you're restricted to just this set of receptors and inherit this from your parents, so it has all kinds of disadvantages. So how the body on evolution actually solved this problem is by creating different components of this receptor. They're called the V, D, and J segment, encoding different versions of those in the DNA. And then during the development of a T cell, it basically chooses one of the 50 V segments, one of the, I think, 20 or something D segments, and one of the, I don't know, 17 J segments, making up the numbers, I don't have them in mind, but something like this, and combines them to make its own receptor. And also at the junctions of these segments, there's like extra randomness going on. So there's amino acids being inserted or deleted. So especially in this junction region, there's a very high diversity, and that's the region that in the end is at the part of the receptor that interacts with the epitope. Sorry. And that region is called the Celia3 loop. So what we have to characterize these receptors is what's the V segment, what is the J segment, they all have names, and with a name you associate a fixed sequence. And then there's this diversity region in the middle, the Celia3 loop. And basically, if we want to characterize a receptor, that's the three things that we want to know. What's the V and J region like, and does the Celia3 loop look like? And that's going to tell us a lot about, and hopefully it's going to tell us a lot about the specificity of the receptor. Right. So let's look at the Celia3 loop first. So that's this region where there's a lot of diversity. You can see this sequence logo that they really are quite diverse. So the size of the letters shows you what proportion of receptors, what proportion of receptors have this amino acid in this position. You can see that there's a very conserved position at the beginning. This is how we define the start of the Celia3 loop. So there's always a cysteine here. And then, especially towards the middle, there's a lot of different possibilities, apparently. So I had hoped at this stage to maybe observe a pattern already there. So that kind of all the suspicious sequences that I'm finding maybe have a similar binding sequence. That would have been a strong hint that they're all binding the same target. Or maybe I call it the amino acids here by their biophysical properties, that I would at least find, I don't know, a lot of polar amino acids in the same position, something like this. But I don't know about you, but when I look at this, I don't really see a pattern like this. I don't really think that there's at least there's not one pattern of all of these T cell receptors. There might be several groups, but I don't think they're all going to, I mean, they don't all have the same or very similar binding sequence. So that's interesting, but it also makes it a bit harder for us. And then what I also looked at is whether we find some other anomalies. So for example, I checked whether certain V and J segments occur more or less often compared to the background. And here I really used frequencies of these receptors in very large data sets, so even more than the control samples that I used here, but there's people who've done extensive studies across millions of people to identify these frequencies of these different receptors. So compared to those frequencies, we really find that 2 V segments and 3 J segments occur a lot more often than usual in our sequences. So that might be a hint also to to some binding patterns, for example, these regions are really important to binding to this protein that holds the peptide. So they're called MHC proteins, and also different people have different versions of these ones. So it may be that this MHC protein is actually what makes you more likely to be suffering from this autoimmune disease. Yeah, so that's an interesting result. Also tried to look at k-mers, so sequences of 3 or 5 amino acids maybe that occur more often in these sequences than usual, and I did find some. I'm not yet sure whether they're very meaningful. Yeah, but there's also an option to look for patterns, right? And since I already said I didn't really find a clear sequence pattern when I looked at the logo, I already said like maybe there's sub patterns, right? Maybe there's groups, classes of T-cell receptors that are very similar, but they're not similar to each other. So I tried clustering them. I did this hierarchical clustering just based on Hamming distance, which is how many amino acids would I have to change to go from one sequence to the other. And yeah, of course it finds clusters, it always does, but you can also see that the differences within a cluster, there's still usually about like 6 or 7 amino acids difference. In a sequence that is only 15 amino acids long. So that means they're still quite different. There aren't many sequences which are very, very similar to each other. Right. Now, we spend a lot of time looking for patterns in the sequences, but there's another dimension that we can look at, right? We can look at single receptors, or we can look at repertoires. We can look at the whole patient, all the sequence that we got from a single patient. And we were comparing those and looking for patterns there. There's actually a very interesting thing that came up. So what I'm plotting here is a multidimensional scaling plot of the similarity of the repertoires. So, sorry. So similarity in this case was again simple overlap of sequences. So how many sequences do these patients have in common? And then the multidimensional scaling just means that take this multidimensional distance matrix that I have, so every distance between every pair of patients, and I try to project this onto 2D but keeping the distances as conserved as possible. That's what multidimensional scaling does. So if 2 points are close on this plot here, they will also close in my original distance matrix. So that means they have different, very similar. If they're close, they have similar repertoires, right? And I think it was quite clear that there are really 3 groups of people here, which I creatively called group 0, 1, and 2. And then my first instinct was to make this Venn diagram, because I thought, oh, maybe they really have distinct sets of T-cell receptors. And you can see that it's partly true. So there are some T-cell receptors which are really specific to a single group. Nobody from other groups has those. But there's also still quite a big set that all 3 groups share. So I was a bit disappointed at this at first. But then I thought, okay, let's see whether we can find a nice representation of all of these clones. Right? What if I plot a matrix? I do put all the T-cell receptors on the y-axis, I put all the patients on the x-axis, and then I sorted these patients here based on the group that I found out through the plot, through this multidimensional scaling. And then I was surprised to really see, oh, there's really these like clear lines, right? Phew, sorry. I don't even have to print the lines where the different groups end. You can immediately see it from the plot. I also put the sequences on the y-axis in the order that I found them through the clustering, multidimensional clustering, because I thought that maybe you can see that, oh, this cluster of sequence is very typical for this group of patients, but that didn't really happen. So for example, this green cluster, some of the sequences are typical for group 0, some for group 1, some for group 2. So not really sure whether the clusters of T-cell receptors mean very much. Oh, I'm sorry, I'm going to have to cough. Right. Let's see whether we can continue. And then I thought, okay, well, this is a good representation. If I simultaneously want to show you the clusters of T-cell receptors and the clusters of patients. But if the clusters of T-cell receptors don't really mean that much, well, can't I find a representation that at least really highlights these clusters that we find in the subjects? Because I think you can already see them here. But, but of course, if I plot it like this, it becomes a lot clearer. So all I did here was rearrange the T-cell receptor sequences so that the ones typical for a certain group are grouped together and colored the same way. I also took out the ones that had quite few patients sharing them and that were not typical for a group. And now I think this pattern of groups becomes very, very clear. And you very clearly see that there are some T-cell receptors that are typical for a certain subgroup of these patients. I hate having a cold when I have to give a presentation. Right. So I showed these results to the medical collaborators who had taken all these samples and done all the experiments, and they were super surprised because they don't know anything about subgroups in this disease. So yeah, this is something that we're still investigating at this point because it seems really clear in this data set. But the question is, does it have some real world impact, right? So we tried to do some analysis of clinical variables, compared them between patients of these different groups. You don't really have to understand what all of these are. Basically, I think this middle row is the most interesting because here it seems that there's some kind of pattern going on that the group 1 and 2 get higher scores than group 0. But the main thing I want you to see here is that with this low number of patients that we have, it's just impossible to see any kind of statistically relevant effect here, right? I mean, if you have 25 patients and then you divide them up into 3 groups, you're going to have very small groups. And that's just not going to be enough to find patterns here. We did the same thing with like variables that we think could be biasing. So for example, the medications that they took, how long they've taken medications before the sample was taken. Because it might also be that the effect that we're finding is just, yeah, again, not disease related, but treatment treatment related, or like based on some different external factor. But again, same problem. We just don't have enough data at this point to answer these questions. But we did get the medical doctors interested enough in this to agree to produce more data for us. So